7 in-class lecture, Human Cells, Cell Membrane. So this is picking up our in-class lecture that my iPad decided wasn't actually going to be an in-class lecture, but was instead going to be a recording. So picking up where we left off in class, we were talking about selective permeability. So in the notes, I'm on letter C, selective permeability, which is on the top of page 5. So let me just zoom to that in the slides. Here's your little review of what we did in class. Boom, boom, boom. Okay. Okay, so we talked about functions of the cell membrane as a unit. The first one we talked about was maintaining an electrochemical gradient. Is that the first one we talked about? And we talked about, and here's a diagram that shows that. So talk about this in a little more detail. What we're seeing here is a cell membrane, and we see, what we actually see is that purple protein that's embedded in the membrane is actually what's called the sodium-potassium pump. And that's the thing that works in a lot of cell membranes to maintain that electrochemical gradient. So what that electrochemical gradient is, and what that means is, it means the proper concentration of ions outside the cell compared to the proper concentration of ions inside the cell. So it's not just concentration of ions in general, but concentration of specific ions. And so cells set those concentrations up very specifically, and then use that setup for various things. So that is, conducting electricity across the membrane is one thing that electrochemical gradient is set up for, and other things. Okay, so that was a little review. We did that in class. Selective permeability is where we left off. Well, sort of where we left off. It's not really where we left off. We're still reviewing here. So we had talked about in class that we had talked about the phospholipids that make up the majority of the membrane. And so we have this membrane, this cell membrane that's made out of lipids that are specialized so that part of them can hang out in water, on the water outside the cell and the water inside the cell. But if something's going to cross that phospholipid bilayer, it needs to be able to pass through the phospholipids unless it's crossing the membrane using a channel or using a transporter, or one of these other methods that we're about to talk about. So selective permeability basically means that the cell membrane is permeable, meaning things can cross it, but it's selectively permeable, meaning that only certain things can cross it, and it can change what it's permeable to by making changes in itself. So anything that is lipid soluble or small or neutral in charge can cross the phospholipid bilayer without an issue. So very small molecules, such as gases like oxygen and carbon dioxide, can pass right through. Those are also neutral in charge, so they're small and they're neutral, so they can pass through the way a breeze can blow through a window, a screen, an open window has a screen on it, can go right through without a problem, but bigger things can't get through that screen. And something that's lipid soluble also can get right through. So for instance, fat soluble hormones are things that communicate with cells, but unlike the water soluble hormones that need to find a receptor outside the cell, lipid soluble or fat soluble hormones go right through that phospholipid bilayer, and they find actually receptors inside the cell that they interact with. So fat soluble hormones are a good example of lipid soluble molecules that pass right through the cell membrane without a problem. Okay, but if something is not lipid soluble or not small or not free of charge, not like free, but if something is an ion and it has a charge to it, or if something is large, or if something is water soluble and we still need to get it across the membrane, now we're going to have to do something to get it across. We have to use a channel or a carrier protein to get it across the membrane, in which case we still can get it across just fine as long as a channel is available or a carrier protein is available and open, because those aren't always all open all the time. So sometimes certain ones are closed, and that would be called a gated channel, and they're only open after they interact with a specific messenger, like a hormone, for instance. Then they'll open up, and then substances can come through, but they might be closed other times. So that depends on the presence of and availability of channels or transporters to get those other molecules through. Okay, so now, so how do cells get substances across the cell membrane? Well, that's what we're talking about right now. But what we're looking at here are other ways that cells get substances across the membrane. So those substances that can't pass through a channel or a transporter, or substances that are not small or lipid soluble or free of an electrical charge. So we have two categories here of ways to get things across the membrane. And the first category are the passive processes. In other words, these are ways that we can get molecules across the membrane where it doesn't require energy to do it. It happens just passively. So some of these things we're going to see, especially in the passive processes, we're going to see things that are going through the cell membrane because they are permeable to it. That would be one of the situations in which a passive process is happening. So the passive processes are ways in which we get substances across the cell membrane, either going in or going out, but a lot of times we're talking about going into the cell where it doesn't require any energy to help that process along. Okay, so diffusion is the first way that things cross the cell membrane, and we've seen diffusion before. Diffusion is just the movement of water across a membrane, and actually, believe it or not, no, sorry. Osmosis is the movement of water across a membrane. Diffusion is just the spreading out of a molecule. So say we have small uncharged molecules like we see here. So say it's oxygen molecules, okay? So they're small, they don't have a charge to them, and they're in high concentration outside the cell. Diffusion is just when things spread out naturally. It's things obeying the law of entropy, which means they go to a place where it's less concentrated. They spread out, they become less organized. So what we see here is the passage of time. As we move to the right, we see the phospholipid bilayer across the middle, also known as the plasma membrane or the cell membrane, and the extracellular fluid is on top of that membrane, or is outside, but that's depicted here on top, and on the bottom is the cytoplasm, so inside the cell. So as time passes, as we go to the right, those small uncharged molecules that we see all concentrated outside the cell in the beginning start to diffuse across the membrane, and they'll continue to diffuse until there's an equilibrium, until there's an equal number of them outside the cell as inside the cell. So that's diffusion. Diffusion doesn't take any energy to get stuff across the membrane. Easy peasy. All right, osmosis, this is the one I started to say that one was. Osmosis is the movement of water across the membrane. So actually water can go right through the phospholipid bilayer. It also can go through channels, and more commonly, and in larger quantities goes through channels, but it can go through the membrane, partly because water is a small molecule, and it's kind of debatable about whether or not it's a charged molecule. It's not an ion, but it is polarized. But it being polarized, it actually is attracted to part of the phospholipid, and so it can attract to part of the phospholipid. It's small enough to slide through the fatty acid part of the phospholipid. So water actually can pass through the cell membrane, but it doesn't pass through, it doesn't gush through or anything. Otherwise, the cell membrane would be kind of pointless. But it can move across the cell membrane. And what water will tend to do is, essentially, this is water doing essentially the same thing that we saw with diffusion. So we could think of this as, on the left, we have water more concentrated on the outside of the cell, and on the right, we have the water less concentrated inside the cell. But really, what the more accurate way to look at this is, outside the cell, on the left of the diagram here, there's a lower concentration of solutes, of some specific thing that is dissolved in the water. And on the right, inside the cell, we have a higher concentration of that solute. So what water wants to do is equalize that distribution. So water wants to flow across the membrane until those dissolved molecules that are on the right inside the cell, that they are as dispersed in the water as they are outside the cell. So the presence of those molecules will attract water in until there's a water equilibrium across the membrane. So that's osmosis. And so that's the movement of water. And so it doesn't take any energy to do that. Water will just flow across the membrane slowly, but it will do it in an effort to equal out the distribution of the solutes in the water. Okay, so that's our second passive process. So the first one was diffusion, second one is osmosis, third one is bulk flow. Bulk flow is the diffusion of large quantities of substances from a region of high pressure to a region of low pressure. In other words, it's essentially like diffusion, but it's a stronger, faster process because because the concentration of a substance is much higher on one side of the membrane than it is on the other side of the membrane. What we're looking at here in this diagram is the respiratory membrane. And so what that is it's the cell membrane of the alveoli in the lungs, and right next door to it is the cell membrane of the capillary that contains the blood that wraps around those alveoli in the lungs. And so there's a high concentration of oxygen in the air inside the lungs and a low concentration of oxygen in the blood. Conversely, there's a high concentration of CO2 in the blood and a low concentration of CO2 in the air inside the alveoli. So that difference in concentration, we could look at either one of those and call it bulk flow that's happening here. So if we just look at oxygen going from where there's a high concentration of oxygen to where there's a low concentration of oxygen, so again, yes, this just looks like diffusion, and it is that, but it's happening at a much faster rate, which is why we call it something other than diffusion, and because it's being pushed along by that very high concentration of the substance on one side and the very low concentration of it on the other side. So across the respiratory membrane, that's an example of bulk flow happening. Bulk flow also happens in cells that are doing a filtration process, like for instance cells in the kidneys that actually have they have more pores in their cell membranes for a filtration process to happen. That would be another example of bulk flow of that happening, but there it's not so much substances going through the phospholipid bilayer, it's going through channels, but still it's a larger quantity of things crossing the cell membrane at once due to a pressure difference on one side or a strong concentration difference on one side versus the other. So that's bulk flow. And then our last one is facilitated diffusion. And all facilitated diffusion is diffusion of substances that actually need to pass through a channel protein to get across the cell membrane. So they can't go through the phospholipid bilayer, but say there are channels and the channels are open and the channels are available, then things will just naturally flow from an area of higher concentration to an area of lower concentration. And that's what we see here. We see an open channel protein, we see a high concentration of green hexagons outside the cell and a low concentration of green hexagons inside the cell, and so they're naturally going to flow. That's diffusion. They'll naturally flow from a high concentration to a low concentration. So that's called flowing with the concentration gradient, and that doesn't take any energy to happen. And in essence, all of these passive processes more or less are that. It's a movement of substances with the concentration gradient. In other words, they're moving across the cell membrane to equalize the concentration of them on either side. So those are all passive processes. So let's go on and take a look at the active processes. So when we need to use energy to get something across the cell membrane, we typically are needing to use energy because we either are pushing something against the concentration gradient. In other words, we're taking like an ion or some substance in the cell and moving it in a direction where there already is a high concentration of it, which is not a natural direction that it would want to go. And so since we are pushing it against the concentration gradient, it's going to take some energy, some ATP to get that to happen. So we're either moving something against the concentration gradient or we're moving larger substances. I think those are essentially the situations in which we need to use energy, larger substances or larger concentrations of substances, where we're moving the whole cell membrane to move something across. Okay, so let's look at these active processes. So the first active process is called the first category is called they're all called active transport. But the first one we're looking at is called primary active transport. And primary active transport is a specific thing called the sodium-potassium pump. We actually saw it in an earlier slide, but this is the sodium-potassium pump. It is a highly specialized carrier protein that what it does is it uses ATP to run it, and it pumps sodium outside the cell and potassium inside the cell. And by doing this, what it does is it sets up this concentration gradient of a high concentration of sodium outside the cell, and a lower concentration of potassium inside the cell. So every 3 potassium ions that get pumped out, what did I say, potassium or sodium? Let me start over. For every 3 sodium ions that get pumped out, 2 potassium ions get pumped in. And so the net change here that's happening is we end up with a high concentration of sodium outside the cell. So that's the sodium-potassium pump. And every time we do that exchange of 3 sodiums out and 2 potassiums in, it takes 1 ATP molecule to run the pump like that. So we can see each of the steps here in this diagram of the sodium-potassium pump. So when the pump is open to the inside of the cell, so it's open to the cytosol, then part of that protein of this pump, or of this transporter thingy, protein, is receptor sites for 3 sodium ions. So the 3 sodium ions will come along. They're naturally attracted to those receptor sites there, and there's also a receptor site available for ATP. So ATP and sodium all bond to the sodium-potassium pump. Once that happens, once the ATP bonds, it goes ahead and does its thing. It separates off that one phosphate, which is breaking the high energy bond, which is transferring its energy to the sodium-potassium pump. That transfer of energy is seen as a change in the shape of that protein. So when that protein changes shape, it closes to the inside and opens to the outside. In that process of opening to the outside, those sodium, the places where the sodium was attached, those areas of the protein, they all change shape just enough for the sodium to lose its affinity to those binding sites. So the sodium is no longer attracted to those binding sites and comes off and goes outside the cell. But also in the changing of shape, potassium binding sites have been revealed. So potassium ions are attracted in, they attach to those potassium binding sites, and once they attach there, that stimulates the release of that phosphate from the ATP, and another change in the shape of the protein. So when it changes shape the second time, the potassiums lose their affinity for their binding site. They end up inside the cell. That the phosphate from the ATP comes off of its binding site, and now the cell has rearranged its back in this, or not the cell, the protein has rearranged its back in the position that it was to start out with, where 3 sodium ions can now attach and a new ATP molecule can attach. So some cells will use up to 40% of the ATP that they produce just to run this sodium-potassium pump. So some cells use this sodium-potassium pump a lot and constantly, and specifically there's big nerve cells and muscle cells that are doing that a lot. Okay, so this is called, the sodium-potassium pump is called the primary active transport. And it's called primary active transport because it's happening, like boom, 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 this pump is running and sodium's getting pumped out and potassium's getting pumped in, and we're setting up this concentration gradient. And with the concentration gradient set up, now other things can happen. So we've run that pump to create this high concentration of sodium outside the cell. Now that high concentration of sodium it doesn't like to be in this place where it's highly concentrated. It wants to go somewhere where there's a lower concentration of it. And so we can pretty passively allow that sodium to rush back into the cell anytime the membrane opens up channels to allow that to happen. And so that happens. That is the flow of electricity across a cell membrane, which happens again in nerve cells and in muscle cells, and it's part of the communication process that those cells do. But also, when that sodium is rushing into the cell, we also will do something, or the cells will do something called secondary active transport, and there are two types of secondary active transport. The secondary active transport occurs as reactions to the movement of the sodium ions. So there's symport, in which the movement of substances across the cell membrane into the cell against the Symport is the movement of substances across the cell membrane into the cell against the concentration gradient by attaching to incoming sodium. So all that means is symport is using that incoming sodium to bring other stuff into the cell as well. So we've done this active process in primary active transport of pushing all this sodium outside, and now through secondary active transport, and this one specifically symport, when sodium comes in, something else can attach itself to sodium. So now we know sodium has a positive charge to it, so a little ion with a negative charge could attach itself to sodium, and it could come in with sodium, and that would be a way that we could passively get that negatively charged ion into the cell. Okay, because the negatively charged ion wouldn't necessarily on its own want to come in, because cells do have an overall negative charge inside, which is what attracts the sodium in and what makes sodium a good thing to use for the sodium-potassium pump to set up that concentration gradient. So we can slip things in while that sodium is slipping in. That's symport. So transporting in, kind of piggybacking off the sodium that's rushing in. The other one is called antiport, which is also shown here. Antiport is repelling things out, also in response to that sodium. So when sodium's rushing in, we could take a positively charged particle that we want out of the cell, and it could repel away from that sodium because they have opposite charges and get kicked out of the cell while those sodium channels are open. So that one is antiport. Here it is. It's the movement of substances across the membrane out of the cell against the concentration gradient. So again, both of these are against the concentration gradient, which is why it would take energy to do it. But it's, the symport and antiport is actually, that's a passive part that's happening in response to that primary active transport that has already happened. So it's being repelled by that incoming sodium. Okay, so symport and antiport, we don't really see those a lot. Big deal, blah, blah. Okay, let's move on. All right, so there are other types of active transport. The other types of active transport are a category called vesicular transport. So all of the types of vesicular transport are using vesicles, transport vesicles. That's why it's called vesicular transport. And those transport vesicles are essentially a little pinched off membrane, either pinched off of a Golgi apparatus or a Golgi complex, or maybe a rough or a smooth endoplasmic reticulum that was, forming some concentration of some substance. And so now that substance is contained inside of a membrane, and that membrane is made of phospholipids, just like the cell membrane is. So vesicular transport is using a transport vesicle to move something into the cell or out of the cell. And these transport vesicles essentially are going to change places or become part of the cell membrane, or are going to be made from the cell membrane. So we'll see what I mean with this. So I think there are 4 of these that we're looking at. The first one is phagocytosis, which you've probably heard of. It's a fairly common term because our white blood cells do phagocytosis all the time. Phagocytosis literally means cell eating. And it is a type of endocytosis. So endocytosis means something going into the cell. So when white blood cells do phagocytosis, what we're seeing here, that yellow squiggly line across the center is the cell membrane. And the large particle that we're seeing above that, and that looks sort of like a kidney bean, a red kidney bean, that's a bacteria cell, okay? And our cell that plasma membrane is part of, that's a white blood cell. So that large particle, that's a bacteria that our white blood cell wants to get rid of. So what the cell membrane of the white blood cell does is it forms what are called pseudopods. So those kind of ridges in the cell membrane on either side of the bacteria, those are called pseudopods. Those are, and it takes lots of ATP to do this and lots of proteins to kind of push this formation of the cell membrane. But the cell membrane changes place, reaches out these pseudopods, and the pseudopods, imagine these two, like the top part of these two ridges, coming together until they meet each other on top of the bacteria cell. When they meet each other on top, they form a vacuole, which is what we see on the bottom of the diagram. So the cell membrane meets itself, and so then part of the cell membrane becomes a vacuole, and then the remaining cell membrane, meets itself across and closes up, so that there's no gap left in the cell membrane. But it forms a vacuole. So this vacuole is this pinched off part of the cell membrane that then will go in a white blood cell. It's going to go and merge with a lysozyme, or a lysosome. Lysosomes, remember, those organelles are the ones that contain enzymes that can break things down, such as bacteria. So it will merge with a lysosome, and those enzymes in there will break up that bacteria and kill it. And that's how white blood cells work. So, phagocytosis is a type of vesicular transport in which we get a larger particle, like a bacteria cell, into the cell. So that's phagocytosis. Pinocytosis is almost the same thing, but instead of cell eating, it actually means cell drinking. And instead of the cell membrane reaching those pseudopods out to encapsulate a foreign cell, the cell membrane actually drops down in, forms like a dimple. And so then extracellular fluid fills that dimple, and then it pinches off across the top and forms a vesicle. So now that vesicle is filled with extracellular fluid. So that's cell drinking, or pinocytosis. So that's another way that a cell can do a vesicle type of transport to get large concentrations of extracellular fluid into the cell. Okay, so that's pinocytosis. Receptor-mediated endocytosis is very much like pinocytosis, but it's specialized and specific. And so the difference here is that there are receptors. So we can see here where the cell membrane is starting to kind of dimple to bring in some extracellular fluid. And in that dimpled part, there are receptors in the cell membrane. And so this whole dimpling process won't really happen until those receptors are all filled. So these are receptors for orange stars, apparently. And once there are orange stars in all of those receptors, then we'll get that pinocytosis action happening, where the cell membrane will kind of dimple inside and pinch off. And then what we see in the lower part of the diagram there is this vesicle that has been formed, and inside the vesicle we have the receptors, and we have the things that the receptors received, and we have extracellular fluid. So we've gotten this substance inside the cell, and so now, depending on what that substance is, this vesicle again will probably merge with something inside the cell and get broken down or whatever. So this is actually how we get LDL, which is low density lipoprotein. That's bad cholesterol. That's how we get it inside cells, because it's not really bad cholesterol. It's something that we do need to get inside cells, because that's how we get cholesterol in large quantities into cells. And cells, we know, need cholesterol to build their cell membranes and for other things. Like if it's a cell that's making like a fat soluble hormone, for instance, it's going to need some cholesterol to do that. So a cell will put up receptors in its membrane for LDLs. Once they're filled up, it will pull that part of the membrane inside, and then boom, there it goes. It has a whole bunch of cholesterol it can use for whatever it needs to use it for. So that's receptor-mediated endocytosis. And our last one, so all three of those were endocytosis, so those were large quantities or large items coming into the cell. This one is exocytosis, so this is something going out of the cell. So exo meaning like exit, outside. So something leaving the cell. So this is the exact same thing that we've seen with those endocytosis processes, but in reverse. So this would be the release of something that the cell has produced. So in the vesicle that we have at the bottom of the diagram here, it's a little bit cut off, but there's a vesicle at the bottom of the diagram that has green circles in it. Those green circles are something that this cell has produced. Let's say this is a nerve cell and that those green circles are neurotransmitters. So now it's time to release those neurotransmitters. They're in this vesicle. The vesicle moves to the cell membrane, merges with the cell membrane, and then opens up. And so now the vesicle is now part of the cell membrane. What had been inside the vesicle is now outside the cell, and that's how the cell releases neurotransmitters. And other things get released in that way, but that's just the example that we're looking at here is neurotransmitters. We could look at hormones or other substances that are released in this way as well. And so anyway, that's exocytosis. So it's a vesicle filled with stuff. That vesicle merges with the cell membrane and opens itself up and then spills its contents outside the cell. So that's our fourth type of vesicular transport. Which brings us to the end of this video. So in our first part of our class today, we looked at the we looked at the structure of cell membranes, which somehow is not an objective here, but it should be. And then we looked at 6 functions of the proteins in cell membranes. We talked a bit about selective permeability and the 3 characteristics of molecules that affect permeability. In other words, whether or not something will be able to cross the cell membrane. Those 3 characteristics being lipid solubility, size, and charge. And then we looked at different passive processes and active processes to get things across the cell membrane. So the passive processes were ones in which substances were moving with the concentration gradient, so they were moving where they naturally want to go anyway, either through the cell membrane or through a channel. And either very passively or faster, like with a push of a high amount of pressure on one side. And we looked at active processes. And in those active processes, we looked at the primary active transport, the sodium-potassium pump, very important in a lot of cells. And then some secondary active transport, the symport and antiport, that can happen alongside the rushing in of sodium that happens after the primary transport, the primary active transport happens. And then we looked at 4 examples of vesicular transport. So we looked at 3 types of endocytosis. So we looked at phagocytosis, pinocytosis, and receptor-mediated endocytosis. And we looked at exocytosis, all examples of vesicular transport. So your homework for this section, this is the last section the first section of class, but here you are getting it so late, but here it is. What I want you to do is draw a small section of a cell membrane like the one shown here. So on your drawing, you're going to show phospholipids, you're going to show cholesterol, you're going to show glycolipids, so you'll show all 3 types of lipids that are in the cell membrane, and then you'll show a protein. And so in this particular diagram, we see a couple of proteins that cross the whole cell membrane. And in this diagram, it doesn't, we don't know what the function of that protein is just by looking at it, but we also see some cytoskeletal proteins just inside the cell membrane as well. So show me some proteins in the cell membrane, and then list out those 6 functions of cell membranes, of the proteins in the cell membranes that we talked about. So that's your homework for this section. Okay, anything else? I don't think so, but so there's this. You have the Bruce Lipton video to watch for your entertainment pleasure. You have the next video, which you maybe have watched already by now, which is on the first part of energy production. And then after that is the second one, the part 2 on energy production, in which we're looking at the electron transport chain. Okay, and then that's it for our week 7. All right, so enjoy the next few videos, and then I will see you in class next week.